Hi, all right, so welcome to this lecture on meiosis and genetic diversity. That's a big part of what meiosis is supposed to do. So I wanted to give its own brief little introduction as to how meiosis promotes genetic diversity. Before we do that, let's go over a couple of really essential parts to make sure that we can talk about this with the actual language that we're supposed to. So number one, let's look at homologous pairs versus sister chromatids. A homologous pair is a pair of chromosomes um, that you've inherited from your parents. One of the pair comes from your mom, and the other pair comes from your dad. These chromosomes are going to have the same genes at the same locations, but they may differ in their alleles. And what that means is that the gene would be considered the category, and the allele is the actual adjective to describe that trait. For example, let's say this gene location codes for your hair color. On your mom's, that would be the gene, the hair color. And then the allele on your mom's chromosome says that you'll have blonde hair, and then on your dad's chromosome, it would say that you have red hair or something like that. So alleles are the adjectives and the genes are the category. So again, homologous pairs are chromosomes that have the same genes but different alleles, one of which came from mom and one of which came from dad. During S phase, each one of these chromosomes is going to duplicate itself and then connect to its copy and create a sister chromatid. So here's the same original mom chromosome that copied itself and then connected through a centromere right here. This is called a sister chromatid. Same thing happened with your dad's original copy. Um, it went through DNA replication, copied itself, and then connected to itself through a centromere, and that's called a sister. That's called a sister chromatid. Together, this compared to this, these are homologous pairs of sister chromatids. Um, Here, if you wanted to get into the chromosome number counting, you have two chromosomes here. One chromosome, two chromosomes. You also have two chromatids. One chromatid, two chromatids. Chromatids are kind of the physical copy, and chromosomes are the genetic story being told. Because this is a copy, it's the same story, so it only counts as one chromosome. But there are two physical copies of it, so you'd still say two chromatids. Same thing here, it's one genetic story being told on these things, so it's one chromosome, but two chromatids. Okay. During crossing over, which happens during prophase 1 of meiosis 1, um, the homologous pair of sister chromatids find each other, and they get next to each other. And a couple of gene segments from a homologous pair literally cross over and exchange DNA sequence. This is so that when the cell divides, it guarantees that the daughter cells will have a blend of your parents' genetics. Oh, just to reiterate, these are really no longer, right here, they'd be considered sister chromatids. And over here, since crossing over has switched them up, they're no really longer identical. Instead, they're referred to as recombinant DNA. Okay, the other method of genetic diversity is called independent assortment. And basically what happens here is um, all of the chromosomes that belong to your mom and all the chromosomes that belong to your dad align in such a way that it's random, so that when the cells are created, when the cell divides and creates two daughter cells, it's a mixture of both cells. So for example, if all the dark colors are from dad and all the light colors are from mom, you could arrange it so that all the dark colors are on one side and all of the lighter colors are on the other side. This would make it so that when the cell divides, these four sister chromatids would go on and be all the genetic information from your father, and these would include all the genetic information from your mother. But what really happens is that these chromosomes are independently aligned or independently assorted to make it a mixture of your mom and dad's chromosomes when the cell divides. And that's how you get this really great genetic diversity. So here's an example of this happening. Um, for a cell with two homologous pairs, there's four possible combinations that can result. So one possibility of meiosis occurs here and will result in two possible combinations. Here's another possibility, which will result in two other combinations. So a total of 1, 2, 3, 4 combinations is possible. That's for a cell that only has two homologous pairs. We have 23 homologous pairs, so to find out the number of possible combinations, you basically take 2 and raise it to the 23rd power. That number is 8,288,608 possible gamete combinations. Add to that crossing over, and you get a tremendous amount of genetic variation. Um, just to give this, let's take a break and count the chromosome chromatid differences. They're very, very similar, and in most cases, they can be used interchangeably. To be very pedantic or to be very specific, a chromatid refers to the physical aspect of a chromosome, kind of how a book is a physical aspect of a story, and the chromosome is the genetic story. In almost all cases, when you're counting chromosomes and chromatids, one chromatid is going to equal one chromosome and vice versa. However, there's one exception that can be made. When identical chromatids are connected through a centromere to make a sister chromatid, you count it as two chromatids because there's two copies, but it's still just one genetic story since they're identical copies. So when you have a sister chromatid with a centromere connecting them, it's two chromatids but one chromosome, and when they split apart, it's two chromatids and two chromosomes now that the stories are going their separate ways. All right, so tying it all together to the big picture, why genetic diversity is good. Um, Evolution is based off of genetic diversity. If you don't have genetic diversity, then evolution can't really take place. If we were all clones and one of us was infected by a virus that killed that individual, then everyone would most likely die from that virus because we don't have the genetic diversity to overcome that or to outlive that virus. So the fact that some of us are varied in our genetics allows us to maybe possibly adapt to that virus or live through that viral attack. Maybe we do have the genetics that aren't going to be damaged by that virus. So that group of people would live on. So the more diversity a species has, the more likely it is to adapt. It doesn't mean that you're guaranteed to adapt, but it means that you're just more likely. There are 4 ways that biology encourages genetic variation. One is crossing over, and the other is independent assortment. Both of these occur during meiosis, which is what makes meiosis such a tremendously important process. Number 3 and number 4 we'll get to in a couple of weeks, so I'm going to keep them hidden for now. Okay, thank you very much. If you have any other questions about how meiosis and genetic diversity are related, please come in and ask me a question.